Okay, so this time on the BioNet seminar, we have Loïc Marrec with us. And he's currently a postdoc in theoretical ecology and evolution in University of Bern in the lab of Claudia Bank. And before that, he did his master's in physics and covering fundamental physics, theoretical physics, and complex systems. And after that, he did a PhD in modeling the evolution and spread of antimicrobial resistance in Sorbonne University in Paris. An interesting coincidence is that one of his PhD supervisors, Florence Bitbo, gave us a talk in one of the previous sessions. And if you're interested in her talk after Loic's talk, you can check on the record on our YouTube session. Okay, I guess that's a brief introduction, and I give the floor to Loic.
Okay, so thank you for this very nice introduction, and thank you for inviting me as well. So now I'm gonna share my screen. Yeah, so can you see my screen?
Yes.
Yeah, okay.
Yep.
So yes, so as Xiangli said, today I'm gonna talk about the evolution of antimicrobial resistance in a changing environment. So it's mainly my PhD topic that I did in Paris with Anne-Florence Bitbol. So yeah, so maybe before I start to talk about my project, I would like to define some key notions, but I'm pretty sure that most of you already know what a microbe is, but still. So what is a microbe? So a microbe is any organism invisible to the naked eye. So it includes bacteria and microfungi, for instance. And to give you an example, in each human microbiota, we have between 10 to the 13 and 10 to the 14 bacteria. So as many as there are cells in a human body. So bacteria are everywhere. And so what is an antimicrobial? So basically it's a drug that inhibits the growth or favors the death of microbes. And so they are very useful to help control pathogenic microbes. And so why did I decide to study?
I'm sorry, Loïc, sorry to interrupt you. Are you aware of the fact that the presentation is not in full screen mode, or is it an error of the screen sharing?
Oh wait,
we see like the PowerPoint.
Okay, so it's not
okay.
Yeah. Okay, so I'm not sharing the good. Wait there. So thank you for letting me know. So okay, I'm gonna go like this. And now, is it okay?
Okay. Yes.
Yeah. Okay. Thank you. Thank you very much. Yeah. Okay. And so yes, why did I decide to study the antimicrobial resistance? Because it's a major public health issue. So since some years we've been observing the emergence of antimicrobial resistance. So the more drugs we use, and the more pathogenic microbes become resistant to these drugs, which is a big issue, right? Because it compromises the treatment of infection. Okay, sorry. And so yeah, and so okay, and so according to a British report published in 2014, the antimicrobial resistance could become the first cause of death in the world by 2050. So it's a major public health issue. And so some questions we can ask are, what conditions can favor or challenge the evolution of resistance? how can we optimize antimicrobial dosage, and what is the impact of antimicrobials on the environment. Okay, so in the following, I'm going to try to answer some of these questions. So my talk will be divided into 3 parts. So in the first part, I will deal with populations of fixed size. In the second part, I will deal with microbial populations of variable size. And in the last part of my talk, I will briefly talk about gradually deteriorating environment. So, okay, so the first step was to build up a model which is based on some experimental observations, I mean, about the evolution of resistance. So first, we know that most mutations giving antimicrobial resistance come with a fitness cost. So it means that the resistant microbes divide slower than the sensitive microbes. Second, this cost can be compensated by a second mutation, and we can assume that the acquisition of resistance is often irreversible. Okay, so here is my model. So in this graph, I plot the fitness or the division rate versus the genotype. And so here in green, you have sensitive microbes that can divide normally without drugs. And as you can see here, a perfect biostatic drug prevents the sensitive microbes from dividing. Okay, so here its division rate is equal to 0 with a drug. And so these sensitive microbes can mutate into resistant microbes, which can divide with and without antimicrobials, but with with a smaller division rate. And these resistant mutants can mutate into compensated mutants, which are very dangerous because they don't have any selective disadvantage compared to the sensitive microbes with and without antimicrobials. Okay, and so as you can see, this fitness landscape is variable. So if you, in absence of antimicrobial, you have a fitness valley, and without no, it's the opposite, sorry. Without antimicrobial, yes, you have a fitness valley, and with antimicrobial, you have an ascending landscape. Okay, and so as you can see, the fitness landscape depends on whether antimicrobial is present or not. And so in the following, I'm gonna address the impact of variable antimicrobial concentrations and the evolution of resistance. And so, yeah, and so since I'm a theoretician, I don't perform experiments, so I had to use a model to simulate populations. So to do so, I used the Moran model. So on this slide, I'm just gonna briefly explain how it works. So you have a population of microbes, and at each time step of the simulation, you select an individual for division and another one for death, in such a way that the population size remains constant over time. Okay, and this model is very convenient because it enables you to compute the fixation probability of each genotype and the fixation time as well. And so for those of you who are familiar with statistical physics, it's what we call a Markov chain. Okay, and so basically to get the fixation probability or the fixation time, we can compute what we call the unconditional and conditional first passage times. Okay. So on this slide, I'm showing you an example of a simulation run. So on this graph, I plot the fractions of the different microbe types, so sensitive, resistant, and resistant compensated, versus time, okay? And so I always start my simulations with sensitive microbes. And so as you can see, at some random times, a mutant appears, but it fails to take over the population, except here. As you can see, this mutant takes over the population. So if you zoom in here, you get this plot. So in the gray phases, I have some drugs, and in the white phases, I have no drugs. And so as you can see, the mutant that takes over the population appeared just before an addition of drugs. So, and so it will be, as you'll see, it will be very important for the following. And so in that case, the process took like 10 to the 5 to evolve resistance. But so if you don't add antimicrobials, resistance evolves through valley crossing, and it takes 10 to the 7 generations. So it's much longer without antimicrobials. And if you add, if you consider a continuous presence of antimicrobials, then the resistance cannot evolve because the sensitive microbes don't divide. Okay. So as you can see with this example, alternations of antimicrobial presence greatly accelerate the evolution of resistance. So now that we know that the most important process in this model is to know when a resistant mutant appears, I would say a resistant mutant appears just before an addition of drugs, we can make some analytical predictions. And to do so, we just need to take into account different timescales. So for instance, the appearance time of a resistant mutant, the disappearance time of a resistant lineage, and the period of the alternation of antimicrobial presence. So for instance, in that case, if you consider a situation where the period is very short compared to all the other timescales, then you can say, okay, the mutant that will take over the population will appear twice the time of appearance. Why? Because maybe someone has a question.
I don't think so. I think you're
okay. And so yes, for this situation, so we can compute the time, the average time at which a mutant appears and fixes. A resistant mutant appears and fixes in the population. And so in this situation, when the period is much longer than the disappearance time of a resistant mutant, but shorter than the appearance time of a resistant mutant, then you can have at most one appearance between two phases with drugs. Okay, and here again, we just need to know when such a mutant will appear before an addition of drugs. So here again, we can compute the probability that a mutant appears just before an addition of drugs, and using this probability, we can get the average time of appearance of a mutant that fixes in the population. And here again, if the period is much larger than all the other timescales, we can do exactly the same thing. So in that case, many mutants can appear before an addition of drugs. And what we want to know is when a mutant will appear before an addition of drugs. And so here again, we can compute the probability and get this time. And so as you can see, we get exactly the same average time for the second case and the third case. Okay, so in practice, we won't be able to distinguish both of them. And so finally, I can sum up my result here. So, and yes, maybe I should say it again. So this time is the time at which a resistant mutant appears and fixes in the population. Okay. And so here here are my results. So in this, so on this figure, I'm showing you the total time of full resistance evolution versus the period. So as you can see, we can observe two regimes. So in this one, the total time of full resistance evolution doesn't depend on the period, and in this one, you have a linear dependence on the period. Okay, and so it was predicted by my equation. And yes, I should say here that okay, I plotted for different population sizes, and the data points are simulated data, and the solid lines are my analytical predictions. So there is a very good agreement between the data and my analytical predictions. And so in this graph, I plot again the total time of full resistance evolution versus the population size for different period values. And here again, you can see that there is a very good agreement between my simulated data and my analytical predictions. And we observe that we have a linear dependence on 1 divided by the population size. Okay. And so when you take a look at these two graphs, you can see that fast alternations accelerate the evolution of resistance, especially for large populations. And so we can even go further, considering, for instance, a case where the duration of each phase is different. I mean, the duration of the phase with drugs and the duration of the phase without drugs. And so, as you can see here, so on this graph, I plot the total time of full resistance evolution versus each duration phase. And as you can see here, we have a minimum of the full resistance evolution when both durations are more or less equal. Okay. And so it's very, it's very interesting to study the case of asymmetric alternations, because you can observe such events. So for instance, in that one, you have a mutant appears just before an addition of drugs, but the phase, the gray phase, so the phase with drugs, is not long enough to make the mutant fix in the population. So you have like this drift over multiple periods. And in this situation, here again, a mutant appears just before an addition of drugs, but this period is not long enough to make it fix. And then the period without drugs is too long. And because of that, since the mutant has a selective disadvantage, it goes extinct. Okay. Okay, so just to sum up on this part, so I show you that alternations accelerate the evolution of resistance, especially for large populations, and we get a minimum in the time of resistance evolution when each phase each phase has more or less the same duration. Okay, and so this has some implications for clinical and experimental situations, because, for instance, we know that the concentration of drugs exceeds the MIC, so which is the minimum inhibitory concentration, for at least 40–50% of the time. So in practice, some treatments may favor the evolution of resistance. Okay. Okay, maybe do people have some questions about the first part, or I should continue? Okay, okay. So in this part,
there is one raised hand actually.
Okay.
There was someone raising.
Yeah, sorry, I raised my hand, but I can also wait till at the end of the talk. If it depends on what the organizers prefer. So.
No, but for us it's totally fine. We are fine when the speaker is fine. So if the speaker likes to get questions,
I'm fine with having questions now.
Okay.
Okay, great. Thanks, Louis. So I was wondering. So I guess what you're saying is if the concentration is varying over time, but I suppose the concentration can also vary over space, right?
Yeah.
So within a patient, I guess you can have different areas that are exposed or not to antibiotics. So how do you think about that in this?
Yeah. So yeah, because in this work, I didn't consider a spatial structure, but we could implement this model in with a spatial structure. So for instance, you could have multiple subpopulations connected via migration paths, for instance. And but it's a very good question. Actually, in this case, so how can I say it? Because we did with San Florence, we had a project about structured populations, and we saw that it's very, so actually how we define the migration asymmetry between each subpopulation is very important and can have an impact on how the population evolves. So I guess it would depend on the population structure you consider and the migration asymmetry.
Yeah, I guess I'm thinking even without migration, that simply because I guess you're assuming that the patient is a well-mixed.
Yes, exactly. Yeah.
Right?
Yeah.
And that's not necessarily the case. So maybe you have areas where basically there would be gradients of the antibiotic concentration itself, even if there's no migration, right?
Yeah.
That you would some would be exposed to higher doses than others, and then
so maybe
that would change.
So maybe I will try to answer at least in part your question with the last part of my talk.
Perfect.
Because yeah, okay. I will. So please ask me again this question at for the last part. Okay. So should other questions or I? Okay, I continue. Okay, so now I'm gonna deal with microbial populations of variable size. Why? Because so in the previous part, I worked with populations of fixed size, but as you know, many drugs are designed to make the bacteria die. So basically, it would be very interesting to take into account extinctions, and to do so we need to relax this constraint on the population size, and so that's why we decided to use another model, and a model of microbial populations of variable size, and more precisely microbial populations following a logistic growth, as you can see on this graph. So with this curve. So basically, a logistic growth is at the beginning you have an exponential growth phase, and then the population size reaches a steady state and fluctuates around the steady state. And so it's very convenient because it allows to take into account extinctions, and also it enables us to include population dynamics and population genetics within the same model. And to simulate such populations, we can use a Gillespie algorithm. And so in the following, I'm going to test two types of antimicrobials. So a biostatic drug that prevents the sensitive microbes from dividing by decreasing their division rate, and biocidal drugs that kill the sensitive microbes by increasing their death rate. Okay, and so in the following, I will try to see if there is a difference between both types of drugs, and also if the treatment succeeds in eradicating the microbial population or fail. So here is what I get for a perfect biostatic drug. So just to remind you, a perfect biostatic drug prevents the sensitive microbes from dividing. So in that case, their division rate is equal to 0. Okay, so no divisions. And so on this figure, I plot the probability of extinction, or let's say the probability of treatment success versus the period for different carrying capacities, or let's say population sizes. And as you can see, we can observe two different regimes. One for the short periods where the drug fails to eradicate the population, and a regime where the drug is more or less efficient depending on the population size. Okay, and so to understand what happens in, for instance, in this regime, so we can take a look at this simulation run. So in this figure, I plot the numbers of each type of individuals versus time. So here again, in the white phases, I have no drug, and in the gray phases, I have drugs. Okay, and so as you can see, the phase with drugs is not long enough to drive the population to extinction. So you have like a drift over multiple periods, and then at a moment you have a mutant that appears and fixes in the population. Okay, and so now if I take a look at this regime, so as you can see here, the phase with drugs is long enough to drive the population to extinction. Okay, so the key point to distinguish both regimes is the time needed for extinction of the population. So this line, basically. And we were able to compute the probability of extinction in this regime. And so, yes, it's this equation, but I'm not gonna detail much. Yeah. And so here on this slide, I'm showing you what I got for the case of a biocidal drug. So as you can see, I get very similar results. But if you compare the value of the probability of extinction, you can see that in the case of the biocidal drug, I get lower values than in the case of the perfect biostatic drug. So the question is, why do I get a difference between both of them? So the answer is this slide. So here again, I am showing you an example of a simulation run. And so as you can see, if I zoom in on this part, a mutant appears while the drug is present. Why? It is possible because a biocidal drug doesn't prevent the sensitive microbes from dividing. So even if it increases the death rate, they still divide. And if they divide, a mutant can appear, okay? And if a mutant appears in this phase, it will have a selective advantage. Why? Because the biocidal drugs are impacting the sensitive microbes. And so that's why I can, here again, I can compute the probability of extinction, but I need to add a new term to this equation to take into account the fact that the mutant can appear while the sensitive population is going extinct. So it is true for biocidal drugs and for imperfect biostatic drugs. Because in the case of an imperfect biostatic drug, you decrease the division rate, but if it is not equal to 0, you can still have divisions and then mutations. Okay, so okay, I can sum up this part. So I showed you that yes, long alternation periods are necessary for eradication, and the perfect biostatic drugs prevent resistant mutants from appearing while drug is present, unlike the biocidal and the subemissive biostatic drugs. But as I'm going to show you here, the range of parameters where the biocidal drug succeeds in eradicating the population is greater than that of the biostatic drug. So here I'm showing you two heat maps. So in that one, I show you the probability of extinction versus the period and a quantity that defines if the drug is efficient or not. And so in the blue area, the treatment is efficient, and in the red one, the treatment is not efficient. And I do the same thing for the biocidal drug, and as you can see, the blue area is larger in the case of the biocidal drug than in the case of the biostatic drug. So it shows that each kind of treatment has different merits. So yeah, do we have questions for this part? Or I can continue.
Is this statement, maybe I have a question. This is a very interesting result. I was wondering whether this statement of the increased efficiency of the biocidal drug depends on the other parameters of the models that are not of the model. Like so how robust this is in terms of the other parameters, which I imagine are not perfectly known experimentally.
So actually, we define two parameters. So the division rate of sensitive microbes and the death rate. And so to make a link between both kinds of treatment, we defined this quantity. So it is the difference between the death rate and the division rate of the sensitive microbes in presence of drugs. Okay, and so it should be robust because because yes, we can make a link between both treatments using this quantity. So, and we only need to define two parameters. So yeah, the division rate and the death rate. So I don't know, maybe you have another.
No. It's I actually for some reason I had convinced myself that there were more parameters, but what you're saying makes sense. They're much less than I actually had in mind.
Okay.
Okay, great, thanks.
And okay, and so maybe now I can move on to the last part of my talk. So yes, so now I'm gonna deal with a gradually deteriorating environment. So in the two first in the first parts of my talk, I assumed that an abrupt change of environment. So I assumed that as soon the drug is added to the environment, it impacts the division rate or the death rate of the sensitive microbes. But actually, it can be a bit more complex. So, for instance, if you take a look at this figure, we can see that the pharmacodynamics is a bit more complex. So, for instance, in this figure, I'm showing you the net growth rate of any sensitive microbes versus the antibiotic concentration, and as you can see, it's very smooth. Okay, and so we can assume that there is a delay between which you add the antibiotics to the environment and the time where it is, and between the time it is efficient. Okay, so that's why in the following I'm gonna change my model. So I'm still considering a wild type, or let's say a sensitive microbe, and now I'm gonna assume that its division rate varies like a sigmoid, okay, and more specifically as a Hill function. So on this figure, I'm showing you different examples. So you just need to define an inflection point, and which is basically the time at which the division rate is equal to 0.5, if you and what we call a Hill coefficient that defines how abrupt the curve is. Okay, so for instance here, if n is very large, then you have like a very abrupt change of environment. Okay, and so if you just consider a population of wild type individuals evolving with such a fitness, you get such a population dynamic. So in this figure, I'm plotting the number of wild type microbes versus time for different Hill coefficients, and as you can see, all of them go extinct. And of course, if the Hill coefficient is very large, then you have like a very abrupt change of environment, so the population goes extinct very quickly. And so in the following, my question will be, okay, can I rescue this population with a mutant, and more specifically with a generalist mutant, which is not impacted by the change of environment, or by a specialist mutant that whose fitness increases over time, like this curve. Okay, and so what I'm gonna try to do in the following is to compute the probability that a mutant appearing at a given time fixes, and I will also try to compute the probability that a mutant rescues the population from extinction. So to compute the fixation probability, I need to make an assumption. So I can, and we can assume that the number of mutants is very small compared to the number of wild-type individuals when the competition is important. So you know, when you have when a mutant appears, we can assume that the number of mutants is very small compared to the number of wild-type individuals in the population. And so using this assumption, I can write a master equation governing the probability of having a given number of mutants at a given time. And which is very convenient, because then I don't need to deal with a master equation with two stochastic variables. So the number of wild-type individuals and the number of mutants. Here, I just need to focus on the number of mutants. And so to do so, I define an effective division rate, or an effective fitness for the mutant, which is equal to its fitness times 1 minus the number of wild-type individuals divided by the carrying capacity. So this term is the demographic pressure applied by the wild-type individuals to the mutants. Okay, and so we can solve this. We can solve this master equation, and because I am able to solve it, then I can get the fixation probability of a mutation that appears at a given time. Okay, so in this figure, I'm showing you the fixation probability versus the time of appearance of a mutant. So for different Hill coefficients. And so, as you can see here, there is a very good agreement between the simulated data, which are the data points, and my analytical predictions. And what we can see is that the fixation probability increases sharply from the inflection point, especially when the Hill coefficient increases. And so, just to remind you, this is the inflection point. So it is when the population size starts decreasing. Okay. So it makes sense that a mutant is more likely to fix if it appears after the inflection point, because then the demographic pressure is lower. Okay. But so in this case, I assume that I know when a mutant appears, but in practice, we don't know when a mutant appears, because by definition, it is stochastic. So the idea is to now compute the rescue probability. I mean, at the level of the whole process. So to compute the rescue probability, I just need to make a second assumption, which is the process of fixation of a mutant is shorter than the time of appearance of another mutant. Okay, so I mean that I don't have several mutant lineages at the same time. And so then I can compute using what I computed, using what I computed previously, I can get the rescue probability. And so here on this figure, I'm showing you the probability of rescue versus the mutation probability upon division for different Hill coefficients. And here again, you can see that there is a very good agreement between my analytical prediction and the simulated data. But you can also see that there is a sigmoidal increase in the rescue probability so like this. And also, the more abrupt the change, the larger the mutation probability upon division is needed to rescue the population. Okay. And so finally, I was also able to compute the appearance time of a mutant that rescues the population. So I used what I computed previously, and so yeah, here again on this figure I'm showing you the mean appearance time of the mutant that fixes in that rescues the population versus the mutation probability. And so as you can see, a mutant that rescues the population appears after the inflection point. And it's coherent with what we saw previously, because a mutant that appears after the inflection point is more likely to fix in the population. And so here I'm showing you, okay, the mean appearance time versus Hill coefficient. And so as you can see, the time of appearance of the mutant that fixes in the population decreases with n. So which is here again coherent with what we saw previously, because when n is very large, you can see that the extinction time of the population is shorter. Okay, so that's why you have a very short time between the inflection point and the extinction time of the population. And so that's why here you can see that the appearance time converges to the inflection time. And yeah, and so then a question I asked myself was, how are the fixation probability, the rescue probability, and the average time of appearance of the mutant that fixes impacted by the population size? And so, as you can see on this figure, the fixation probability doesn't depend on the population size. So here I have some data points for different carrying capacity values, so different population sizes. And as you can see, I get exactly the same results for for every carrying capacity. And then here you can see that the rescue probability and the mean appearance time of a mutant that fixes in the population only depends on the parameter carrying capacity times the mutation probability. Okay, so the fixation probability does not depend on the population size, and the rescue probability and the time of appearance of the mutant that fixes depend on population size only via the product k times mu. Okay, and so yeah, maybe I can sum up this part. So we have quantified the fixation probability of a mutant and the rescue probability of a population destined for extinction, and this contributes to the understanding of evolutionary rescue. And the faster an environment degrades, the more difficult it is to rescue. And so yeah, here I applied my approach to the question of antimicrobial resistance, but it is valid for any time-dependent fitness and all time-dependent wild-type dynamics. And so it's very interesting to study climate change, increasing antimicrobial concentration, etc. And I think, and so to conclude this talk, I would like to thank Anne-Florence Bitbol and Raphaël Voiturier, my PhD supervisors in Paris, but now Anne-Florence in Lausanne, and Claudia Bank, my postdoc advisor, and my current team here at the University of Bern. And thank you for your attention. And yeah, I will be very happy to take questions.